Okay, so I think we will go ahead and get started here, and we'll obviously let anybody else drift in as they need to, but I'll go ahead and get started and introduce everybody. So welcome everyone to the final session of this year's Go With The Flow expert webinar series. Today we have brought back all of our panel experts to have an in-depth discussion regarding two key topics. The first will be Process Analytical Technology, or PAT, Insights and Challenges, and then the second will be Future Perspectives of PAT and Manufacturing Biologics. Today we are joined by Jay West, Principal Scientist at BMS, who presented biologics concentration measurement using inline flow VPX instruments during the UF-DF process in drug substance manufacturing, showing the progress that his team has had in implementing the inline flow VPX into their downstream UF-DF for drug substance. Next, we also have Renata Pocitarenco, Associate Scientist, and Marius Mueller, Team Lead for Analytical Fill Finish Support from Janssen Pharmaceuticals. Their presentation, Automation of Biologics Drug Product Manufacturing through Modeling and Flow VPE. There they shared how Janssen is forging into new ground with model predictive control and how they plan to implement PAT and automation into manufacturing using MPC. And just this week, a couple of days ago, we had Aishwarya Ramanan, Senior Research Scientist from Seagan, who presented implementing PAT strategy using Flow VPX system for inline monitoring during batch and continuous chromatography. Aishwarya presented her perspective on all of the potential opportunities for the Flow VPX system in continuous manufacturing process and work being done in various stages of the purification process. Our expert moderator for today is Ralf Kuriyel, Senior Vice President, Research and Development here at Repligen. Ralf joined Repligen in 2016 as the Senior VP of Research Development, where he oversees the company's R&D efforts, focusing on innovative solutions to address biologic development and manufacturing needs. The structure for today's panel discussion is simple. We'll break it into a couple sections: PAT Insights and Challenges, and then Future Perspectives. For each section, we will ask the expert panel predefined questions to get the conversation going. Throughout this discussion, at any time at all, please ask your questions using the control panel on the right side of your screen in the GoToWebinar platform. Click on the question tab, and there you can enter your question. At the end of each of these sections, we'll pan the audience for questions and ask all the questions that we've received. Remember to feel free to write in your question at any point in time, and we will be sure to get to your question before going on steps. So without further ado, I want to get this discussion started. I'm going to pass it on to Ralf to kick it off with our Go With The Flow expert panel discussion. Thank you. Ralf, the floor is yours.
Thank you, James. Good morning, good afternoon, everyone. So I'll start with the first question to the panelists. Perhaps we start with you, Jay, and we go around. On what problem were you trying to solve through Flow VPX? And what particular product attributes of Flow VPX made you think about using it for the problem you're trying to solve?
Sure, that was my question, correct, Ralf?
Yeah, please go, and then we'll go around for this one.
Sure. So the so-called problem that we were trying to solve, going back about 5 years ago during our drug substance development at Bristol Myers Squibb, was that our biologics, particularly the monoclonal antibodies, the drug substance concentration target was going from 50 to 150. That's a massive obvious increase in drug substance concentration. And the reason we were doing that is for subcutaneous injection, which is a much simpler way of administering these medicines. So it was a really kind of quantum leap in terms of the clinic and delivery of these. However, it presented us like a significant manufacturing challenge, especially in terms of when you're reducing the volume of your drug substance during this operation, and you're relying on weight and the change in weight during the operation, it can be quite inaccurate, and you can overshoot, you can undershoot. So there was no real way to reliably control the process without monitoring the concentration live inline. And so that's what happened is we purchased some of these a few flow VPE instruments and started using them in our process development and pilot scale labs. And yeah, so that was the problem, is that we had no control or idea, of that, what the accurate concentration was during this process.
Thank you, Jay. And how about you, Aishwarya?
I think I'm very much in line with Jay and what they saw. So throughout my career with downstream, I've seen instances where we've had a challenge in optimizing our, say, the TFF step. So I think I've always wondered it'd be really nice to have an inline concentration measurement system so we can complete our TFF with better accuracy. And that's where it started. And then when I started doing more and more chromatography, I realized, say, for a step like a DBC, we're looking at a large number of samples that need to be read offline, and also having to wait for the results, and so the wait time in general, and also the amount of sample that you're using up to determine your DBC. I felt it would be really nice to have an inline control or an inline real-time measure for steps like those. And also, I think for most part, the UV detectors max out with any chromatography system. And I think those are times where I felt a variable Pathlength technology would come in very handy.
Thank you. And how about Marius and Renata? How about your perspective on it?
Maybe Renata, I can start quickly. So for us, it was similar also, because we were also at some quite blind spots in the process. And I think that's basically where we started off in the whole PAT initiative that we were looking into, to really bring also the fill finish and the drug product process to really have like online technologies that would enable that. And while mapping this drug product fill, we were also looking at where can we use, or where can we replace several CQAs that we are currently taking samples. If we go into and analyze them in a lab, in a QC environment, where could we potentially replace those ones and see? And eventually we were going out and saying the most critical one, or like one of the most important one, is definitely protein concentration. And we were actually going out and looking to what kind of technologies are there, and found like things from terahertz, IR, Ramanan, and so forth. But eventually ended up with the flow VPE back then, and found this to be a good and actually quite promising technology that we then basically went forward to, especially also that the technology was the same, also using A280 compared to current offline technologies, which use the same, which made it easier also to adopt and also to go forward with this one. And yeah, I guess with this one, really it helped to basically increase shift, helped to increase sample count that you would have. So basically it came with all the benefits that these online technologies have.
Yeah, thank you. And Marius, in your, I'm sorry, please.
No. Just to add to what Marius just said, the dilution process for which we build our control feedback algorithm is one of the most important steps in drug product manufacturing, and in fact it counts for up to 1/3 of the products manufactured in particular at the Schaffhausen site. And with that, having the possibility to measure protein concentration, it came naturally to think about a way to automate this the dilution process, which with a concentration measurement, it's easy to think of.
Thank you, Renata. So in the presentation that you guys gave, Renata, Mario, there were a few slides. You talked about validation of the methods, accuracy, precision, and so on. How did you go about your thinking pattern in terms of validating it and making sure that the tool was indeed the right tool to do the job you had and to be within the specification of your process?
So this actually was, as I said before, was also in line with the offline technologies that we were using. So it was really like, I mean, current technologies, we have the Solo as well as A280 as comparator technologies. So therefore, we basically applied similar criteria and specifications that we also had for those, and then really went forward. And as you mentioned, really, so we really looked on the one hand definitely on specification, like on specificity, that this was basically working. Also then range for a certain product, including intermediate precision. But again, for us, what we had seen, especially having this new technology, it was most important to have an offline comparability, also showing a head-to-head with the current standard, which is usually, I think, not part of classical method validations, but due to the as this was asked quite a lot, we were basically also having this data included in all the validation works we did.
Thank you. And Aishwarya, you applied it to different, you talked about TFF, you talked about chromatography, perhaps inline concentrator. How did you go about this validating the method and so on?
Yeah, so for me, again, like Marius was saying, I think comparability was what I was looking for starters. So we had the VPX measurements compare with the Solo, which is our typical offline measurement system. And also for something like titer measurements, we compared it with pretty much the platform out there, which is the CDX BioHT. So comparability was important for starters. And I think we are looking more for repeatability, as well as I think the robustness will be the parameters that I would like to validate.
Thank you. Renata, you in your presentation, you talked about deterministic models and coupling with machine learning for good adaptive control. And how did you integrate the models and the control, the way you went about it?
In the algorithm we built, there are a few contributors. So we have the main part and the core part, that is the feedback loop, which basically learns something about the system through the measurements, and based on these measurements and this knowledge of the system, calculates and estimates an optimal action to write back to the system. So as I was explaining in my presentation, there is this complexity of the time delays in the system, and for that we evaluated the possibility of having this time delay quantified given the system physical properties and characteristics. But this is not a reliable thing to have for this time delay concept, because it can vary due to many factors, as I was mentioning during the presentation. One of these easy to think about is, for example, the scale of the process, and the scale of the process can greatly impact the time delay. So aiming to quantify the time delay with experiments, it's something that it's really not worth doing, because it couldn't provide sufficiently good estimation of this time delay in new processes. For that reason, we went for a data-driven approach, and it was a different way of thinking to this time delay in our system. So considering the measurements provided by the FlowVP and what we expected to see thanks to our mechanistic model, we found a way to make our data-driven algorithms learn while going and while the new measurements were acquired in time.
That's great. Thank you. Jay, you go ahead, please, Jay.
Yeah, I wanted to ask a follow-up question to Renata. So this time delay, is that solely a function of a mixing issue with the buffer addition and before it reaches equilibrium?
Yeah, the time delay is a function of many things. One of these is for sure the mixing. the mixing efficiency that can be impacted by the impeller speed, impeller characteristics themselves, the fluid properties, but also other factors like where is the flow VP placed in our recirculation loop? How long does it take the solution to go to the flow VP? How long does the measurement take? So many things that could be quite difficult to quantify and to measure experimentally. So for that reason, we went for the data-driven approach.
Thank you.
Yeah.
Jay, you did a lot of PD work like all of us, right? And then what key insights did you get during your process development work that drove you to push the technology towards GMP?
Yeah, so thanks, Ralph. So I put something in the chat. I'm not sure if others can read it, but something else came into my head that sort of answers this question and the first question you had about the interest in the flow VPE, VPX and PD, is the Donan effect. I don't know if people are aware of the Donan effect, like exclusionary, basically the excipient drift. Yep. Especially above 150 grams per liter, you get significant excipient drift, which in our case, like we published, is histidine and sucrose, where when you're getting in the 200 gram per liter range, you can see reductions of 20 to 30% in the concentration of those components. Yeah. So, because you can imagine, it adds another layer of criticality that you want to stop your process, your UF2 set point at a consistent concentration, because if you stop at, say, at 220 one time or 200, that can make a very significant difference in the concentration of your excipients in that drug substance pool. So that was actually added to the importance of being able to go, I guess, like basically to push the drug substance to the limit in terms of the highest concentration you can go to without causing, precipitation and total loss of CQAs for the protein. And it seems like that limit is around the low 200s range, and especially in terms of viscosity, you can't really push these things beyond that point. But by using this inline meter, we can basically safely and consistently have a process for a whole platform of these MABs at high concentration, and without risk of, like I said, up to the CQAs of these proteins, because we're constantly monitoring the concentration. So that's really critical, I think, in terms of like putting it into the GMP space, that so that we don't have to spend so much time, of course, like validating the process, that and process consistency, because we'll have this thing that's actually going to assure process consistency.
Thank you, Jay. Aishwarya, in your talk, you showed the whole downstream process and all the different places you could use flow VPX. What do you think about the different milestones that are needed to use it more for continuous manufacturing, be it inline concentrator, multi-column chromatography, cell retention technology for perfusion, and so on?
So I think this is a spot where adaptive control would come in very handy. And again, reiterating the points that I mentioned before, moving forward with a technology like this and moving forward with continuous chromatography, I think most industries are slowly stepping into continuous by moving every step of the process, so basically semi-continuous to fully continuous. And I think adaptive control would come in very handy with this transition. And also, I think for starters, from PD, scaling up would be the very beginning before it actually goes any higher or any scaled up.
Thank you. And Marius, you pushed the technology to different sites, IND, BLA. How did you go about justifying the project and the investment? Was it return on investment? Was it risk management? Could you comment on that?
Actually, it was a multitude of those, right? As outlined in the beginning. So first of all, I mean, we're checking what problem we could solve, right? But now that it comes really with, on the one hand, definitely also saving hands-on time, right? Which is usually a big driver when you say you can basically have less time that you need, for instance, in an analytical lab over something that might be done in the shop floor. but also we basically had the samples that are not taken anymore, right, from a material perspective, as well as also the soft benefits, which would, for instance, allow now to do shifts, which would not require to have shifts anymore, maybe in a QC environment. It would also, and that's also one of big drivers, is to reduce then the hold times for instance, for IPC testings, which could be, depending also on where and what, take up to 24 hours, 8 to 24 hours for just the whole time that you would need to include to wait for this result and to have that. So I think at least for the drug product space, the case was quite evident. Pushing into the drug substance space, they were kind of similar, and also the vaccine space, there we had similar drivers where they were saying, yeah, already, you know, I mean, it was already now when they saw already what you can see now during the process, and having this process understanding that they did not have before already was quite convincing. So this usually helped them to create the case further down.
Yeah, and talking about the BLA, the INDs, what kind of evidence did you have to give to the regulatory agencies for the use of technology and relying on the technology?
So on this one, it was mostly the question that most of the time came was how can you have this comparable to what we have? And there we basically used a certain, like a stepwise approach, right? Where we were first creating data from a process monitoring perspective, basically getting a data package there, and then moving it down all the way to really having it as a certain replacement for tests. And the rationale we usually had was, okay, we always showed also offline data, so we always had a certain comparability down the process. Again, what we discussed also before was, as we could refer to the gold standard, the offline data that we also collected during the whole process with it, basically constructed like really a compelling data package. And also we included data we had from our lab scale studies that we did, as well as all the runs that we had at full scale. So this was really the combined effort which eventually led to pilots.
Thank you. And then we're talking about risk mitigation and so on. So from your perspective, Jay, how did the technology help you reducing risk?
Yeah, so in the pilot lab, it's reducing risk because they have a somewhat, I don't know if it's a novel approach, I would say, but along with Marius was saying, like, how do you prove, you know, that this is not adding risk to the process, especially in terms of accuracy. And the way we have gotten around that in the pilot lab is that the flow VPE is used, or an X now, for process monitoring. However, the solo VPE is the measurement of record in the batch record. So basically, once you've reached that target concentration with the VPE and they manually stop it, you know, it allows it to recirculate for quite some time, then pull a sample and then test it offline with solo VPE. So that's actually, I think, the wedge in the door that to get this into a GMP space is to say, yeah, exactly, like we want to use this for process control monitoring, but then next we want to use it to decide when to stop the process to take a measurement. So that there's actually precedent, like in some of our PAT or other PAT instruments, that they're used in that way to basically decide when to stop and pull and take a measurement. So, which is a baby step, but it's getting there basically to the point where eventually, yeah, we can trust these instruments to basically do everything on their own in a sense.
Thank you.
Sure.
And Aishwarya, what are your views on that risk dimension?
I don't think we're there yet, thinking all the way there yet.
Yeah, it's and Renata, Marius, anything on your side or any further comments on risk?
Yeah, so I would really say, so if you really talk about PAT, which would really be to be in control of the process, to really basically have a certain possibility to react on unforeseen things, right? And that's basically what we now do, right? We do see now how the process is, how the process reacts, and this is really something, and I guess Renata can definitely speak on that when it comes to the dilution process, that you can really still interact, and you still can stop the process over the process is done, and then you take a sample, and then eventually you have the risk already. But this comes also, I guess, with various stages where you see mixing homogeneity. How long does it take in order to really have homogeneity of a certain batch? Getting insights into the whole that, I think this really comes with that. And yeah, the PAT in general plays there a really critical role to be able and be ahead of, rather than acting but also reacting on things.
Thank you.
Totally agree with Marius. It's exactly what we are trying to do with our automated dilution.
Thank you. James,
Okay.
I think, sorry.
Jay has his hand up. So
Yeah, I just want to add one other point for the really potential use for the VPX that we did not foresee is that after the drug substance is done, the formulation is completed, it's pumped into bags and frozen. And it happened to notice actually that when you're using these high concentration processes, that the concentration in the bags are not consistent and can even vary by 10-20% per bag. So it basically, because of some sort of, like a settling factor or whatnot, basically. So in the future, I think it would be quite valuable, and we have some ideas on basically to put a VPX in line with the bag filling operation to ensure that the concentration is consistent in the frozen bags, because that's an issue right now.
Thank you, Jay. James?
Oh, yes, thank you so much, Ralph, and thank you guys for the discussion. We've had just a couple questions come in here that I just wanted to toss everybody's way before we go on to the next segment in our discussion. The first question that I've had come in is, it seems like everyone is using VPX for various applications. Are all of you using it in the manufacturing stage as well at this point, or is it all in pre-manufacturing?
Maybe we go around, we start with you, Jay, because you're in the panel, and then we go to Marius, Aishwarya, and Renata.
Sorry, can you repeat the question?
Sure, so I'll abbreviate the question. Essentially, the person asked, is everyone using this in their manufacturing stage as well, or is everything currently being in stages?
Okay, I think I understood the question. Yeah, I trailed up there. So when they say manufacturing, I think they mean GMP, correct? So
yeah, I'm assuming that. Yeah.
Yeah. So we've been using the pilot scale for quite some time, which is not GMP, but it's a 1/4 clinical scale, and we are in the process of tech transferring it into the GMP space, so not quite there yet.
Okay.
And then how about you guys, Marius, Aishwarya, Renata?
Yes, we use it in definitely in the manufacturing space. So GMP manufacturing space, from clinical to commercial, and as said, right now more and more also the drug substance, as well as for vaccines as well. They are all manufacturing scale. However, we also have use of it in a lab scale to really also accommodate there for studies that we might eventually run on a lab scale. But yeah, so all scales is the simple answer.
Great.
Thank you, Marius.
I think we're still in the research and developmental stage, so we're still researching more uses with the VPX.
Okay.
And maybe
Great.
I can say something about the algorithm, the control algorithm we have, and it is not in the manufacturing yet. We are working on bringing it there.
Thank you, Renata.
Thanks so much. So another question that did come in was actually asking about method transfers and challenges associated with that. So I think, Marius, it sounds like you guys have done it. Did you have any challenges associated with transferring methods with the VPX to the different groups?
Yes, we did. The issue we have is, I think, again, having the nature of rather something that is not an offline technology where you basically have an instrument where you can basically say, look, this is the instrument, that's how it's used, that's basically how it's used everywhere, actually gave us quite a challenge, because it can be used depending on how the setup will look like for various sites, how it would look for there, how it would look there. Actually, we needed to be quite creative there, and also seeing how we can leverage things, right? How we can have things that we might do on a lab scale, how we can have this then on engineering scale, or basically on a full scale, and also seeing how the equipment differed, how the process would differ, to also account for this as well during the transfer.
Sounds good. Thank you, thank you. I think we got kind of time for one more question before we go over to our next segment here. So the question kind of asks, how are you guys working with your QC groups to align on with required CQAs that they do offline? Are all these offline measurements still needed? And are you still pulling samples, or do you also envision ever not needing to pull samples in the future? So I guess we'll kind of go in the same round table section. We'll toss it to Jay, Marius, and Renata, and Aishwarya.
Yeah, well, specifically in the GMP manufacturing space, we so as I mentioned, the flow system is not enabled, excuse me, yet. However, its cousin, the Solo VP, is being used in analogous fashion for basically real-time forward processing on the floor. We do that in some of our commercial processes, but not yet in our clinical processes, and that's something we want to move forward to, basically because it saves several, I mean, I hate to say this, several hours per sample that needs to be tested, because the QC method for high concentration samples relies on what's called a gravimetric dilution. It's a very laborious process. And the feedback we've gotten from QC actually is that they're happy to have us take this away from them, because it often involves like somebody working a third shift, because this process typically ends late at night. So if someone has to be there late, you know, you need all these people to sign off. It's very stressful for them. They don't like it. They definitely prefer this is something that we take off their hands. So that's what the feedback we've gotten from QC.
Great. So that's good, Marius.
It's also, so as mentioned, so during the process, we would still keep the samples to really get like a head-to-head comparison for as long as needed to really get the data package. But once we are there, right, and we have the finding, then we would basically realize, so that would be then also the benefits to not have QC measuring any samples anymore, to not have these. Nevertheless, we still have it as a backup, as a potential backup in case something would basically fall out or basically would not work during setting this up during any of the manufacturing.
Thank you, Marius. Is there one more question, James, or shall we move to the next section?
No, I think we are all set there, Ralf. So I'll let you kind of kick off the next section here.
So it's future perspectives. We'll talk a bit about digital twins, Industry 4.0, and then real-time release. So in terms of Renata, you did the model, machine learning, deterministic model, and so on. What do you see the role, can you comment, of FlowVPX in creating and moving toward that direction of having digital twins and stronger adaptive control?
Yes, so definitely the FlowVPX can play a huge role in moving forward towards these directions, because it offers insights into one of the most critical attributes, as Marius was mentioning before, that is the concentration. So whenever I would say that whenever we have the availability of this real-time data, we should try to build models, build a deeper understanding of our processes, and try to move into a more objective future where we only rely on what we see in the process at each time point, and we take actions based on this knowledge, this real-time knowledge.
Thank you. And Marius, from a Janssen perspective, other comments from for digital twins industry 4.0? Is that a direction that you guys want to go more and more?
Yes, definitely, and I think that's exactly. I mean, if you compare like the pharmaceutical industry to other industries, right, where you have a lot more of this automation, a lot more of digitalization, I think that bears also kind of really challenges for sure on the one hand, as Renata outlined, but I think also gives us some chances there, right, that we can still build them at now and build them up and also see how we can have them. Where I think, again, the PAT really plays a central role in that, really have also technologies that would enable this, that would able to react on those, that would, by having it combined with any mathematical modeling, with any artificial intelligence, to really, again, react rather on processes than only monitor. And I think this is really where the whole thing is going and where the whole thing basically needs to go. But it needs still also the digital infrastructure, which, again, I think is in the infancy compared to others. And this is really something to basically align. And as we really talk about PAT, integrated quality, real-time release, I think they always come hand in hand with a proper digitalization and a proper digitalization strategy as well.
Yeah, that makes sense. And the North American perspective from your side, Aishwarya and Jay. Let's start with you, Aishwarya.
Sorry, could you repeat the question again?
So any thoughts, your perspective on FlowVPX and the role it could play in creating digital twins and moving towards Industry 4.0?
Yeah, so I think, yes, definitely the VPX will be a very good instrument in terms of automation. And I think pretty much every step of the process would or may benefit from automation. And I cannot unfortunately speak too much about it at this point because, like I said, we're still in the developmental stages. But I think it will definitely be a promising way to go.
Thank you. And Jay?
Sure, yeah. My perspective on the VPX is it's a trailblazing technology for enhanced PAT that will serve as a model eventually, I think, for how we, and this is kind of the industry consensus too, because the VPX is based on the Solo VPE, which is based on A280, all very proven technologies. That this will be like the first of the really enhanced PAT methods that makes a significant impact in the process, and from which all future PAT methods, will basically follow that template. And it's a multi-stage evolution. I was just looking at the white paper we did. Basically, it's a 5-step process. It's not going to happen in a day. It could happen as quickly as probably as a decade, the full digitization of the process. But there's a number of issues I think we need to. They're not significant issues, but they need to be addressed by, I think, the most senior people in biopharmaceuticals. Things like review by exception. That's a big one. Can a computer review a result and basically for a process? Is that, because QC always checks it several times, reviews it before it signs off. All electronic batch records, that kind of thing. Basically, obviously ensuring that all that data is stored safely and securely, and of course that will enable what's called investigations. That's one of the great parts of this, is that it's really going to make everything much more consistent, and you'll be able to pinpoint where problems occur much more quickly. But yeah, there's basically several steps that we have to get before, especially with the technology. We need more inline instruments that can measure CQAs like in a very high precision, basically, so they can do this inline, release and basically say, okay, this is the quality is sufficient and it's ready to go.
Thank you.
But yeah. So we'll probably best case scenario is a decade. We could have like a plant that's fully operational. We're releasing drug substance in a week.
Thank you, Jay. Marius, Renata, you guys built that nice model. You're using it for control. Do you think it will help in the journey towards real-time release?
Yes, I definitely think so. First of all, we should align on what real-time release means. As Marius mentioned, during our discussion. And in fact, in our with our automated dilution model, we cannot really finish the dilution and go immediately to the market, because there are other steps after the dilution that need to be taken. But the automated dilution can definitely shorten the time of the overall drug product process. And as I was mentioning during my presentation also, an estimate of the time currently for doing the dilution process is around 6 to 8 hours. And with our new vision, we could reduce this time to 1 to 2 hours. So it's a big improvement in time terms.
Yeah, indeed, it's very impressive. So in terms of the progress for real-time release, perhaps we go around the panel, and we chatted a bit previously as well. And Jay, you made some comments. How do you see the progress towards that? Mario, do you want to comment on that, Marius?
So, yeah, I would say that basically, not. I mean, as Renata said before, right? It's really something where we would need to say, what would we say about real-time release, right? And if we say real-time is a driver, I think there it is great to have this, but. And I think this is like the flow VP is one technology that we might have, but I think there are still more to this than that basically would eventually enable a real-time release. And I think in a pre-discussion we were also talking about things such as BioBurn, which is currently, I think, one of the tests that takes the longest before having something released. So if you basically would manage to get this size down, or basically if you have the time much more quicker, well, then other assays are in the spotlight. And I think you can then really just go through that to see which one is then your roadblock to really enable real-time release. And I mean, if you would talk about an ideal case, we would say real-time release should be once the last vial is filled to be able to have the batch ready and released. So this is, I think, the ideal case. But I think on that way, it's, I mean, if you can already shorten time, if you can really already bring that much earlier, that is definitely helpful on the way there.
Thank you, Marius. Aishwarya, Jay, your perspectives on the journey to real-time release?
I think like Marius and Renata were saying, I think when we're talking about real-time release, I think we're going to have to define what that exactly means. And like they were saying, I think there are many parameters that go into real-time release, like product quality, bioburden, endotoxin. There's so many characteristics that go in there. And I think every one of these would play a major role when we're talking about real-time release. And I think when we have a proven way to show putting all this together is, I think, will be a challenge going forward.
Thank you, Aishwarya.
So I have in order to, I've sort of already laid out, I think that there's a 5-step process, or at least that's what we envision to get to full real-time release. And the pressure I see will come from potentially 3 different areas, and particularly in pricing, because biosimilars are no doubt already will become much more common, and that will, you know, the cheaper you can make the drug, obviously, this, and you know, the real-time release is going to decrease the cost of operations and manufacturing. Another one is the advent of continuous manufacturing. There may be drugs, like already I know in our pipeline, that cannot probably be manufactured reliably without continuous manufacturing, just because, you know, this you have to have continuous harvest, because certain CQAs will go out of spec if you do a typical batch process. And the third one is contract manufacturing organizations that are manufacturing biologics. They need to tech transfer in products very quickly and get the line running and manufacture as quickly as possible to obviously maximize profit. So I'm more like, like companies like Lonza are already incorporating a lot of PAT so that they can have basically a platform like hopefully, or eventually maybe real-time release process where they're manufacturing drugs to their customers quickly and efficiently with minimal time for tech transfer.
Thank you, Jay. And the last question, and maybe just a few words of advice, all the learnings while you were working with Flow VPX, what would be, and we'll go around again, starting with you, Jay, your advice to someone who is interested and thinking about it on the use of Flow VPX based on your experience. So let's start with you, Jay, and then we'll go around the panel.
Let's see. So, so it's a simple. Excuse me. It's a relatively simple instrument when you, and it can be platformized, but you do have to be very careful in some ways, and in some of the settings in order to basically achieve like maximum precision and accuracy. And that definitely needs to be tested. And you really, I think, need to understand the technology and how everything works. And because that for example, too, like the Pathlengths, the VPX are getting so short that there's potential. I don't want to slander the product at all, but you can have protein sticking to the fibrate, which can change the accuracy under certain conditions. So it's really something that you need to, I think, to test thoroughly with all your products to ensure that it's giving you accurate readings under all the conditions that you desire. Because you don't want to just. There was actually an issue earlier on with some of our VPEs, because as we know, they replaced the VPE with the X because the need for higher accuracy above 150 grams per liter. So we had, our scientists that were not analytical just using the instrument without doing offline testing to see if the comparability was there. So they're just using it without doing the proper, testing, comparability testing. So anybody who uses it, definitely, make sure you do all that so that you don't get poor quality data.
Yeah, do your homework and make sure everything it makes sense. And Marius, let's go to you, and then we'll go on to Aishwarya and to Renata.
Yeah, I would say definitely. So for us, it's basically, if I had known what I know now, like 6 years ago, I mean, we would have had the process much faster. So it's really also like, there's quite a lot during the process to really implement that, because as said before, it's not something where you have an analytical tool and you do basically the analytics there, you basically do the method validation, development, and then place it there. No, it's much more complex to this, sadly, employing their people. And I think this is really like something I would give people along, is to have a clear roadmap and also make a proper mapping of stakeholders, right? Because we have seen, for instance, that cleanability is a big thing. Then also having the whole engineering and manufacturing there, also have it with people from the shop floor so they know how to operate, to train them, to have like the full package around that. I think that is quite a challenge that we might have not foreseen before, but well, eventually do nowadays.
Thank you. Aishwarya.
So yeah, the VPX is definitely a more straightforward system in its mode of operation, and I think the user interface is also very user-friendly. And having said that, I think to understand or to implement the instrument's capability to full use, I think you're going to have to do a bit more digging into how the instrument works and the full capabilities of the system, and to figure out where exactly in your process would it fit, and what kind of data or the sweet spot for your data generation, I think would need more study into it.
Thank you. Renata.
And yeah, from my side, first of all, my advice is to make sure to learn. I worked with the Flow VPE, and my advice is to learn which are which is the order of the steps to follow in order to not break the cuvettes, because this happened to me a few times. And besides this, to pay attention when someone is working with variable concentrations to these time delays and therefore to make sure to interpret correctly the measurements when there are variations in the concentration. because it may be that you need to wait a little bit in order to see the right concentration in your process.
Thank you, Renata. James?
Awesome. Thank you guys so much for the great discussion. Again, it looks like we have some time for some questions that have come in. And the first question that I see here is, you guys have mentioned that flow VPX is being used at various different points in the bioprocessing sequence and everything. How many systems do you guys envision yourselves using or are using, and can this be instantly moved to different points in the process? I'll toss it to Jay, Marius, and then Aishwarya and Renata to go around.
Yeah,
this.
Sure, Matt. I'd like to have as many as possible, right?
Sure.
Yeah. In every, well, to be frank, honestly, I think for downstream operations, if you had, you know, in a perfect world, you would want to track the mass throughout the whole operation, right? And that could be done with the VPX, of course. You know, you would get a very accurate mass to time, you know, plot basically from your all the elutions off your chromatography operations and during UFDF. But realistically, I would say, you know, first, of course, the low-hanging fruit is the UFDF. That is, I think, where you're going to get the most bang for your buck for, especially because the material is completely pure, so we know the accuracy is there, and that really is where it shines and is needed, especially for high concentration operations. So there's a couple, like as I mentioned, actually, I'll share a little tip. We've thought this through because even during the UFDF operation, like it would be nice to have it after recovery for during dilution and then maybe for backfilling. So instead of having 3 instruments, one way I visualize this could go is that you would, you could, and especially if you don't want to mix your product streams, is to switch flow cells because they're making use available the single-use flow cells. So you could have one instrument and just swap out the flow cells as you move along with the process. So you could have the push with the cart to, like, say, the polishing operation if you want to monitor that, you know, like I said, for the flow through, and then move it to the UFDF just by swapping out these single-use flow cells. So that's a possibility.
Thanks, Jay.
Sure.
Thanks. How about yourselves, Marius and Renata? Any insight there on the question?
Maybe for those who saw Renata in my presentation, right, we were also mapping on where would it make sense, or where basically is typically concentration measured for drug product, for finished process. And we also highlighted that there are different kind of applications where I could think of, right? I mean, it usually also comes a bit on the business case, because we're saying that I think the most benefit we have at the moment is really using it for the dilution, even then controlling the dilution process. But one could also consider to have it maybe after the filter, right, to also see what the filter impact might be, how much of the filter is absorbed. And then lastly, also directly at the point of fill, where it really fills, to also monitor the consistency during that. So there are multiple ways where one could apply it. It always just depends, I guess, also on the business criticality and also on the business rationale.
Good. And Aishwarya, did you have anything else you wanted to add to this topic?
Yeah, so I think, like I shared in my talk a couple of days ago, I think a VPX can go pretty much in every process step of the downstream. I think, having said that, I think you can definitely toggle the systems around if you're to have multiple systems. And I think the way to go would be probably to identify which step of the process is more complicated and definitely would benefit from an offline measurement system if you want to keep it simple.
Sounds good. Sounds good. Thank you so much. So this question is, I guess the person directed over to Renata, but I think anybody else could potentially chime in and answer as they feel fit and necessary as well. So they wrote, aside from modeling protein concentration, are you looking at other NPC models that you're looking to work on? What kind of would be you envision to be the next bottleneck that you'd want to kind of tackle?
So, well, I am not working anymore with the FlowVPX now and the FlowVP now, so probably Marius could answer this question a bit better.
Sure.
Maybe on top of it, right? Because I mean, having this for the control, I mean, there are other technologies, and I mean, Jay, as we talked about, also being part of the Bioforum, there's a lot also on the Raman technologies and the Raman field there, where it's also a lot also on having more chemometrical models that eventually could help throughout the process and so forth. So I think this might also be something where this might apply and definitely help out there. I would say this might be one of the examples.
Right. Sounds good. Sounds good. I think one last question here that we have that's come in. So the person wrote, bioburden still seems to be a relatively large obstacle that we all face in the process and going through to do you see any new technologies or solutions that will help us get around this get around the bioburden process and testing and get us through some of these?
I know people have been working on it. I'm not intensely involved in it, but there are some clever methods. I know, like say that I remember one from years ago that used the fluorescence, the native fluorescence of like say bacteria, which is very common to as like an inline bioburden sensor. I'm not sure how far that that type of technology is advanced, but it definitely seems that it's something you could validate out, right? Or it's like obviously they'd like to do the plate streaking and all that, but it's so time consuming that maybe in parallel for several years you could run your inline meters for checking for bioburden, and then run your plates, and then once it's established that is a valid method, then we can make that switch. But I think it's possible, yeah. And another thing too is like say during drug substance manufacturing, there's several sterile filtration steps further down in the operation. So, if in the, so that's basically, it's a very, how to put this, careful process. So the risk of, I'm not sure how often they fail bioburden, but I would doubt it's too often during the process. So should be able to get there, I would hope.
Great, thanks, Jay. Did anybody else have anything they wanted to add to that question? I think, Ralf, I wanted to quickly turn it back to you before we wrap up. Did you have anything else you wanted to discuss before we end the seminar?
Yeah. We certainly will keep it. It's at Repligen. We're really passionate about pushing our automated systems, marrying it with the proper PAT tools, and with the goal of really giving solutions that makes manufacturing less expensive, and all the goals that we share. I also want to thank you guys, all the panelists, for this very rich discussion, for your presentations, and looking forward to future interactions. Thank you.
Thank you so much, Ralf, and thank you to all of our wonderful panelist members here. I think we had a really great discussion for this hour, and I hope all the folks that attended found all of the discussions to be very insightful. So I hope everyone has a great rest of the day, and that will be the conclusion of our webinar. Thank you, everyone, so much.
Thank you.
Thank you.
Thank you.
Thank you very much.
Bye.
Pleasure.